Hello everyone, and welcome to today's live broadcast, Methods to Account for Sequencing Artifacts in Large High-Throughput RNA-seq Data, presented by Joseph Paulson, PhD, a research fellow at Dana-Farber Cancer Institute. I am Kristen Peretgentiel of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button on the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Joseph Paulson. I will now turn the presentation over to him.
Thank you, Kristen. Hi, everybody. Happy to be here today. As Kristen said, I'm going to talk about a particular instance in our approach for accounting for various sequencing artifacts in large high-throughput RNA-seq data. And so right now, currently, I'm in the Quackenbush lab at Harvard School of Public Health and Dana-Farber. And I came to this lab to learn how to do network analysis, which is becoming much more common with these large high-throughput sequencing data sets becoming available. And I stole a few slides from him on how not to do network analysis. And once I explain how not to, I'm going to explain the approach, the pipeline that we developed to normalize and do quality control of large high-throughput RNA-seq data so that we could get good, robust networks out. So many people often take RNA-seq data. They might have two types of phenotypes, healthy, diseased, or wild type and knockout. And they do a statistical comparison, and they subset on the set of genes, and they get differentially expressed genes, those that happen to be associated with phenotype 1 or those associated with phenotype 2. They take a protein-protein interaction network and they start coloring the nodes of that protein-protein interaction network, and they generate what we refer to as biopoetry. So one question is, should things that are differentially expressed be connected? Why is that necessary? And is the PPI network even relevant? Another common approach that we don't recommend is where you do the same statistical analysis. For example, you might use DE-seq or EDGE-R or a t-test to find the differentially expressed genes. And you'll also subsequently, using all the data, estimate correlation networks. So then you'll you'll color the nodes of those correlation networks again and do your biopoetry. So one question is, are things that are correlated functionally connected? And are correlations the same in different phenotypes? Why are you aggregating all the data to perform these correlations? So the way that we do network analysis and that we recommend is you do your statistics separately on the two different phenotypes when you infer your networks. So you infer a phenotype-specific network. And from each of those networks, you analyze the network topology and structure. You compare the network topologies, and you simultaneously can compare the differential structure and expression. And this can give a more phenotypic specific network and one that you can make more sense of. hopefully. And so in the lab, we've been developing a lot of different regulatory network methods, including one called PANDA, and we released recently on Bioconductor a package called PANDA-R that allows you to estimate the gene regulatory networks, which are different from co-expression networks. I won't get into that today, but we had a number of various network approaches and methods that we wanted to analyze, that we wanted to tackle a lot of open questions with RNA-seq data, but we needed, so here's the PANDA-R bioconductor package, but we needed a big sandbox to actually play around with our methods and test a lot of our hypotheses, biological hypotheses, and see how well our methods worked on large data sets. So we found a big data set, a big sandbox called the Genotype Tissue Expression Pilot Analysis, GTEx for short. And what it was it was a big sandbox in that it had over 450 genotyped donors. There were approximately in the what's called dbGaP data, 9,500 or so samples. So these are over 450 individuals that had around 50 or so, 54 tissues sampled, and the transcriptome from each of the tissues was sequenced. So we've been working quite a bit to analyze this data using our own techniques. And a number of the papers have been either they're in review or they just recently got accepted. And I'm going to touch on a few of them today, especially the processing and normalization pipeline. And you can go to some of these tiny URLs to find the bioarchive, the preprints for these papers. This one is tinyurl.com/GTEx01. So I'll write that real quick. Very well. tinyurl.com/GTEx01. And there you'll find the bioarchive and preprint available to read in case you have any questions for this presentation. Or you can email me, of course. So if we go ahead and we look at Gene Expression Omnibus on the internet, we can download quite a few data sets of RNA-seq data. However, the majority of them, here we have a histogram where the y-axis, it's the frequency, the number of samples. We're presenting the number of samples in each data set from GEO. And the majority of data sets in GEO have less than 50 samples. Okay? Now we have a data set, this GTEx cohort, that has 9,500 or so samples. And a lot of the methods that have been developed for analyzing transcriptomic data have been developed with these sample sizes in mind. So it begged a number of questions. A number of questions arose with this beautiful large resource. First off, what is the quality of this particular data set? And how do we go about looking at the quality? How similar are different tissues to each other in healing? That's one of the biological questions that we had. What's an appropriate way to filter RNA-seq data? And how do you appropriately normalize the data? That's a very important question because you put in two samples. Let's say you have the same exact sample. You put in sample A to sequencer at time 1, and then you put it in again at time 2, you're going to get two different outputs. Hopefully not too different, but you want to you want to normalize the data so that sample A and sample B are comparable. So our solution is YARN, yet another RNA-seq package. And it's the preprocessing and normalization of the GTEx. It was developed for preprocessing and normalization of GTEx expression data and other data sets. And if you just want to go and grab the normalized GTEx data, you can just go to this URL, right here. There are 35 lines or so, and it will pre-process and run all of the commands so that you have great quality data to go and ask your own biological questions. It's currently a bioconductor package that became recently available, and you just need to install the software, and then you'll be able to do everything that we're talking about. So basically, the yarn method is a 6-step workflow. Just like you do 6-minute abs every day and you get a six-pack after a couple of months, right? You follow the 6-step workflow, and in no time whatsoever, you'll have perfect data ready for beautiful figures for your manuscript or analysis. I apologize if some of the images don't come out very clearly, but you can download the PDFs of this entire presentation and it will all look okay. So here we have the preprocessing and normalization workflow that I'm going to describe. And the first step is getting the data. Sometimes that can be quite a hassle. I won't go into some of the struggles we had with dbGaP, but that is oftentimes an issue. Hopefully none of you ever have that issue. If you, like I said before, if you go to this tinyurl.com, preprocess GTEx, you can go and grab the data, at least the publicly available version. So grab the data. Step 2, you want to do some quality control and filtering of samples. You want to filter samples that have poor annotation quality. For example, there might be mislabeled phenotypes for the different samples. There's a paper that recently came out in F1000. Whose sample is it anyway? Widespread misannotation of sample in transcriptomic studies. And they identified in GEO that there were apparently 46% of the data sets had at least one mislabeling. And this was of the sex, the annotation for sex, whether a sample was from a male or whether it was from a female. And so along the way, we might see some code in red, and those are just it's just some code that you can use from the yarn package that will allow you to generate figures like this one, like what I'm presenting. Here, what we can do actually, what we decided to do is to take PCA, it's principal coordinates analysis, and take a set of control genes. So it's a very simple concept where you take a subset of genes that you know will be descriptive of a phenotype. For example, we can take the Y chromosome genes, and I don't remember which tissue this is, but you can subset to a particular tissue. It's nerve tibial, and we subset it just to the control genes, Y chromosomal genes, and we did a PCA plot. And what we observed was that all the females clustered on one side of the PCA, and all the males clustered on the other side, except for one particular individual, GTEx IL-10. And you can do this for all the different tissues, and you'll see that in the GTEx cohort at least, it was very clean in that they had only one individual that was consistently misannotated. So actually, when we put this paper up on bioRxiv, we actually received information that using the PCA method did a good job as in terms of quality control. We were told by the Broad that actually GTEx IL-10 is known to us as a patient that received sex reassignment surgery. So this one simple approach it's very necessary because misannotation, mislabeling is widespread in a lot of transcriptomic studies. Oftentimes we find as high as 10% of the samples, for example in TCGA, might be misannotated. And so this is really just the first step when doing high throughput, when using high throughput data, doing some analysis is to do the quality control filtering. All right, the next question is about merging tissues. So there might be multiple conditions that are separately labeled, but if you could combine them, you might get increased power. You might have initially very small sample sizes for different tissues or conditions, but if you were to aggregate them all, you could have a reasonably sized study. So one particular question, we can use PCA yet again on perhaps the most variable genes, those that are hopefully delineating one condition from another, one tissue from another, and to see that, for example, I apologize, the colors aren't really coming up, but here what we have, let's look at the middle figure on the top panel. Let me use my arrow, this one, and then this one. So if we look at the first one, we see that there is, on the left part of the plot, we have cells. So these are fibroblasts. So they were designated in one of the labels as skin, even though they're just transformed fibroblasts. And then on the right-hand side, we had skin that was sun-exposed, suprapubic, and sun exposed, lower leg, and sun unexposed. And when you look at the most variable genes and you do a PCA, you can see a separation of the cell lines, of the cell line from the tissue of origin, the progenitor tissue. But when you look at just the skin samples, as in the sun-exposed, sun-unexposed, you don't see any separation between the two groups. So perhaps one way to do this would be to analyze all of the cells separately, and then to analyze the sun-exposed, sun-unexposed together. You can combine those two conditions, those two groups, as one. In other instances where we looked at blood vessels, we saw that there were three really distinct groups of the aorta, coronary, and tibial. So just things to consider if you perhaps want to merge samples or conditions for increased power. Our exploration actually led to a result that was surprising for us, that cell lines under tissue of origin differ quite a bit. Here on the lower right plot, we see that whole blood and LCL, whole blood, LCL samples, they were perfect on the first axis, principal component separated from the skin samples, the skin and the fibroblasts. And on the second component, the cell lines separated out from the tissue of origin. So I'll talk briefly about that at the very end, about that result. So that's great. Once we've decided what tissues to actually merge, we have to consider filtering genes, because there are sometimes noisy measurements that come through in the sequencer. But you don't want to filter too much, because there's some tissue specificity. When we began analyzing this data, someone had suggested that we filter anything not present at some level, some minimum threshold in half of the data. Unfortunately, when you're dealing with many different conditions like this, that's going to remove many different tissue-specific genes. For example, and there are a number of examples that we see in box plots here. These are just some that I randomly chose to highlight. And so it's important to consider these thoughts. For example, what happens when you look at just the most variable genes, let's say the top 10 or top 15 of blood vessel, of samples of, just looking at the samples, blood vessel samples. What we see is if you do too harsh of a filtering, you lose out that tissue specificity. Here we have a dendrogram, and the coloring at the very top represents the three tissues. We see the tibial mostly aggregates together, but then the aorta and the coronary tissue samples aren't really separated out. But when you preserve the tissue specificity that's there, we see 3 distinct groups in our clustering. And these are just of the top 10 most variable or 15 most variable genes. So these are just things to consider when you're doing your analysis. So once we've gone about there, our last question really is about normalization. Okay? And there are a number of different approaches in normalization, and I'll get to that in a second. But just to rehash, so we started off with close to 9,600 samples. We had 55,000 genes, 54 tissues. And through the process, we either threw out a number of samples due to quality control, we threw out genes that were very lowly expressed. The details of that are in the paper, in the preprint, and in the Yarn software manual. And to this point, now we have 9,435 samples, 30,000 genes, and 38 tissues. So we've been able to aggregate a number of these tissues for increased power, for example, for EQTL analysis. So now we go on to normalization, a very important part where we have sample 1 and we have sample 2, and we want to make the 2 samples comparable. As I was saying, there are roughly 2 normalization techniques. One is where you have a scaling. One is a scaling approach. For example, I don't recommend this, but you divide by the library size or by the sequencing depth. How many reads are there? So that you can get a proportion of, see what proportion a gene is in a given sample. That is one type of approach. And there are many different approaches used by DE-seq and HR. One is called RLE, TMM, and other library size. Those are called scaled normalization approaches. Other approaches are global distributions, where there are different types of assumptions, and you're not dividing by a constant, you're not dividing by the median count or some other value. Instead, you're transforming the distribution. So let's look at over here in the top left corner of this plot. Let's look at this plot just to get a quick understanding of normalization. Here, what I'm displaying these are 3 different data sets. Each column represents a different data set, but just imagine for a second that this is what we have here are each line is the distribution of counts. Or here it's actually log 2 PM values, but let's just imagine it's the log counts. It's going to be, each line represents an individual sample, and the x-axis are the values. So for example, you might have a gene that's represented that has 10 reads assigned to it. Another gene that has 200 reads. So 200 would be here, one read would be here, and how many genes that are represented at 10, there are quite a few, or the majority of the distribution is there. Now what we see here with all of these samples is that they follow a similar-ish structure, but that there's a lot of variability between the different samples. Let's take a look at this example where there's less variability, there's some, but it's much more tight. And if we look at the colors, we see that the conditions also tend to be different. We see that there are a number of samples that are orange, and we have separately a number of samples that are green, and that there's this distinct difference in how the distribution looks between those conditions. Whereas in this first plot, we had 3 conditions. Okay, we had asthmatic, smoker, non-smoker samples. And we could not really distinguish between the samples, between the distributions based off of the phenotype or the condition of interest. And then here on the far right, we have even more samples, and we see that the green are brain and the red happen to be liver, and they have very distinct patterns. Okay, so this is just what it looks like without normalization. There used to be a technique that's become more popular with a statistical method called VUM for differential expression analysis. That is quite powerful, especially when you have large sample sizes. So once you have more than 100 samples, you probably want to move away from DE-seq2 or from HR and work with VUM. And the type of a normalization approach recommended there is quantile normalization. And what that does is it forces all of the distributions onto one similar reference distribution that is taken from the average of the quantiles. So you define this reference as the average of the quantiles, and then you force each of the samples to follow that same frequency, or that same distribution. So one issue with this method is great, but the assumption is that the variability in the distributions of all the various samples comes about solely due to technical variation. Okay? And that you can control that variation by forcing them all onto the same distribution. What happens here, though, what we observed was that there were two conditions that had very distinct distributions, and that was a biological artifact, not a technical artifact. At least we believe so. So there's this new approach that just actually got accepted yesterday. Hicks et al. Biostatistics called smooth quantile normalization that will do quantile normalization when there is no distinction between the phenotypes, and it will do it separately, or it will do scaling. It will do it basically distinctly when there is a difference between the two conditions. And so here we see brain and liver, for example, plotting onto the two, or defining their own distributions, in essence. Okay. So just to go back to the GTEx data, what we observed here is that there are distinctions between there's a lot of variability, first off. We have a lot of samples, there's going to be a lot of variability between the different samples. Not only that, what we're observing is, let me draw an arrow. We see that this tail at 0 is increasing. You see the density, the spike mass, there's a spike mass at 0. There are a lot more tissue-specific genes. So as we're adding more and more tissues, we get a lot more zeros because there are a lot more genes that are tissue-specific. And the distributions are fairly distinct from each other. They don't look too much so initially, but once you add more and more, it becomes fairly clear. And using quantile normalization in this instance would not be appropriate. And so when analyzing the GTEx data, one should probably do smooth quantile normalization, the Q-smooth. Here is a close-up. Apologies for the mess up there. Here we're plotting the density. Here's the density, here's the expression of the distribution for all the testes samples in the GTEx cohort and all the artery tibial samples. And you see once we zoom in, we see these distinctions, the distinct transcriptome distributional profile for each of the two tissues. We see it very distinctly, and also the variation. What I'm not showing you guys is that scaling normalization approaches like what DE-seq or HR use are not going to be enough to control for that variability between samples. And so by using a quantile a smooth quantile normalization, we're actually retaining the biological profile of these two tissues while also controlling the technological variability there. We can ignore these top two plots. Instead, let's look at the bottom two. What happens also with quantile normalization, here are two examples where here on the left we have no normalization, and we see TBX20. It's an important gene that's known to be in the heart or expressed highly in the heart, and we observe that in the raw data actually of GTEx. These are box plots of this particular gene of the expression, and we see that it's highly expressed in heart right atrial and heart right ventricle samples. And it's not so highly expressed in most of the other samples. However, once you do quantile normalization, this can actually introduce errors. Because we're defining this reference as an amalgam of all these different tissues, the expression for TBX20 shot down to almost non-existent in our heart samples. But for some reason, in other samples, it shot up and became very highly expressed. And that's just an artifact of the normalization technique. And so some people need to be aware that these are issues to consider. So the last point on the normalization is that you can look at the RMSE, the root mean squared error, of each of the individual samples to the full reference. So what we have here on the y-axis, we have the root mean squared error, so the difference between the sample's transcriptome and the reference, log transformed and everything, the log difference. We have the root mean squared error, and we have for each of the different tissues. So we have for each of the different tissues, we have the box plots. And what happens when we define a full reference, just like what is done in quantile normalization, we often see that there are high valued root mean squared errors. Okay, the lower RMSEs, the better. Now, when we define a tissue-specific reference, okay, for each tissue, and we do quantile normalization on that tissue, for example, we get much lower RMSE. And that's one example of one of the advantages of doing something like smooth quantile normalization, the newer approach. So what we just discussed were 5 steps, or a number of steps to actually go about getting the data, performing high quality filtering, okay, merging tissues so that you could get increased power for whatever analyses that you might want to do, filtering genes to get rid of noisy measurements, but while retaining tissue specificity or condition specificity, whatever phenotype of interest that you're looking at, and then of course normalization to make sample A and sample B comparable, to then do your differential expression analysis or to develop your gene regulatory networks. And the last step, the final step, the most fun step, is really to do an analysis. And a number of different analyses that we have done so far have included cell line transformation, looking at sexual dimorphism, looking at the difference in tissue-specific regulatory networks between all the different tissues, doing EQTL tissue-specific networks, understanding how cis and trans EQTLs make up these networks and whether there are tissue-specific effects or global, and a number of other questions that we've tried to address, and they're all available on bioRxiv as preprints if you're interested. I'll just briefly talk about one of them, talking about the transcriptional landscape of cell lines and their tissues of origin. So if you remember back to the PCA plot that I made over here, so in the bottom right-hand corner, we have on the y-axis PC1 and on the x-axis PC1, on the y-axis PC2. And what we observed was that cell lines and the progenitor tissues are quite distinct from each other. And I think most biologists and people know this, but to what extent, I don't think that was well understood. And so we have a computational approach for actually understanding what pathways and what genes are. In a high throughput manner, we finally were able to understand a little bit better these questions. So what we did was, this GTEx cohort is great. It's a big sandbox. We did a number of comparisons. We did differential expression analysis, comparing whole blood and LCL, right? The difference between LCL cell line and the whole blood tissue of origin. And of course, these whole blood and skin, they're convolutions of different cell types, but this helps us understand just the difference between those. Then we took the analysis and did skin and fibroblast samples, and similarly, whole blood and skin and LCL and fibroblast. And in total, there were 89 paired samples. So from the same individual, we had 89 samples that had all four. There were 244 paired samples between skin and fibroblast, and there were 127 paired samples between whole blood and LCL. So we did two types of analyses. The first one was differential expression analysis, where we generated our lists of differentially expressed genes using that technique I mentioned earlier, VUM, which is available in Lima. And we then performed pathways. We looked for pathways that were differentially expressed using gene set enrichment analysis. We did gene regulatory, and then we analyzed separately the network topology of the gene regulatory networks using that PANDA package that I was mentioning to understand key transcription factor regulators, looking at the transcription factor degrees, and to understand which pathways were differentially targeted, looking at the gene degree. So a lot of the results actually made sense, but as I was saying, this is a nice computational approach to really investigate what was known beforehand. So some of the pathways that are differentially expressed between cell lines and the tissues of origin, a lot of them made sense. Let's see, there was well, you can look at a number of them, but for example, cell cycle is more expressed in is a pathway that for the differentially expressed genes that are more associated with, they're more enriched in cell lines, for example. So some pathways that were enriched for at least two groups. In terms of our comparison, we had immune signaling that were enriched for the blood and LCL cell lines. That makes sense. Cell cycle and DNA repair just being enriched in the cell lines. Extracellular matrix pathways that were enriched in the skin and fibroblast. And is cell-cell junctions also enriched in the skin fibroblast pathways. And then what we did after we did the gene set enrichment analysis and the pathway comparisons, we looked at the transcription factors and the genes to understand how what the differences were between them. So like I said, we took the LCL samples and we built a network. Separately, we took all the whole blood samples and built a network. Also, and then we compared the topology between the LCL and whole blood. In a separate analysis, we then did fibroblast gene regulatory network, and we did skin gene regulatory networks, and we compared the topology of those two networks to understand. So what we could do is we took the gene in degree difference, so understanding what transcription factors were associated with which genes, and we took all the genes associated with cell cycle and various pathways, and to show that there were lower there was lower gene in degree for a number of these pathways, for a number of these genes associated with the cell cycle compared to other non-cell cycle pathway genes. So with that, I really want to thank a lot of the people that did these analyses with me. Camila Lopez Ramos, doing cell line transformation. If you go and check out sexual dimorphism, Joey Chan, Stephanie Hicks, smooth quantile normalization, Maud and John Plattig, EQTL tissue specific networks, Avi, Marika, Kimmy, doing tissue specific regulatory networks. And with that, I also have to thank my boss, John Quackenbush, at the Dana-Farber Cancer Institute and Harvard School of Public Health. And thank you.
Thank you, Dr. Paulson, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. It looks like we have a couple of questions to start with. Our first question is, where is the manual for the Yarn software?
Right, so okay, great. Thank you very much. Yeah, so the manual, and I might as well answer the second question also at the same time. Being a part of Bioconductor, Bioconductor is a R repository for various statistical software packages, similar for like Yarn and QSmooth, the smooth quantile normalization package, or PandaR. What you can do is you can actually go to bioconductor.org and you search for Yarn. What you'll see then, you'll go to the website and you will see a home page. Once you get to the Yarn bioconductor package, you'll see this screen, okay? And to install the package, you just have to run these two commands in your R session. After you do that, right below this, there's something called a vignette, and you can either type in vignette, close brackets, quotes Yarn, and that'll open up the Yarn manual. But that's only after you've installed and loaded the Yarn package into your R session. So to install it, don't forget, you have to go to this URL and find it. The URL for going to directly to Yarn is, let me type this in, bioconductor /packages /Yarn. So that will take you directly to the website for it. Thank you.
Okay, well, if there are no more questions, then I would like to once again thank Dr. Paulson for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. And before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August of 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.